Hey everyone, today's guest is Dr. Alessio Lana, whose research group recently posted a preprint showing that immune rejuvenation dramatically extended lifespan, with some mice living close to 60 months, a duration that is unheard of in mouse lifespan studies and achieved or coming close to being achieved by only a couple of interventions, including calorie restriction. Today, Alessio will present those data followed by Q&A. So welcome, Alessio, and let's get into the presentation.
Thank you very much for the invitation. Our lab investigates what we call telomere transfer biology, which is some piece of tremendous biology we've been studying for, I'd say, about 10 years now. And it started when we asked a very simple question, essentially, how T cells are protected from aging and senescence. So all the bad things that come and happens to the lymphocyte as we grow older. So the T cells stop reacting and protecting ourselves from challenges, infections, cancers, and so on. The dogma in this field for decades has been that T cells are capable upon immune activation of turning on an enzyme, which is called telomerase. It won the Nobel Prize in 2009 because it protects telomeres, the end of chromosomes, from shortening. So at each cell division, the cells eventually erodes part of their chromosome ends, and telomerase, what it does, it replenishes that sort of loss. So it adds back about, let's say, 100 to 200 base pairs, right? So people in the field, including myself, for decades, have thought, enzyme, okay, we have what we have to do. We have to, preserve telomerase activation in order to preserve T cell lifespan, okay? But a couple of evidence, I would say, in the field that were suggesting that this was not enough. Because, for instance, if you do simple examples and experiments, when you transduce telomerase in T cells and you culture T cells, they eventually also become senescent. So suggesting to us that telomerase alone maybe was not enough to protect T cells from senescence. So we asked the question, what happens before telomerase is activated? So what happens at the very onset of the immune response? When a T cell encounter another cell type, which is called antigen-presenting cell. These antigen-presenting cells exposes pieces of viruses, pathogens, antigens from cancer cells, so that the immune response can begin to protect ourselves. And what we showed a couple of years ago was that in certain circumstances, an antigen-specific response would lead to this unprecedented sharing of genetic material, whereby the antigen-presenting cell transfer telomeres packaged in vesicles at the neurological synapse, and we called it telomere transfer. So these telomeres then eventually get into the T cell, and by doing so, they trigger a stem-like memory phenotype. So that T cell, before it even engages in the first cell division, it is protected from senescence, okay? So that's the new piece of information that we shared in Nature Cell Biology in 2022. But still, there were other questions. Even at the time, reviewers were asking, okay, but how does it really work? So you say that there is antigen presentation, but effector memory T cells, you're showing that these T cells are not very capable of undergoing antigen presentation, are not very capable of going telomere transfer, despite the fact of being, of course, antigen-specific cells. And the missing piece of the puzzle was that, and we hadn't shown it before, T cells not only need antigen presentation to trigger telomere transfer, but they need to be in a specific metabolic configuration in order to be, I would say, we call it telomere acceptors. So in other words, only if T cells can engage on fatty acid oxidation, this telomere transfer can occur upon antigen presentation and immune synapse. So it leaves essentially naive T cells and memory T cells being capable of undergoing telomere transfer. And this capacity is eventually lost with age as our metabolism shifts towards inflammation and glycolysis. So this is very important. And it explains why only certain T cells among the antigen-specific populations of T cells are capable of undergoing telomere transfer, okay? Then, of course, over the years, I have a company, I'm CEO of Centicell. We've developed different ways of tuning these reactions. One proprietary ways is through the small molecules called DOS that destroys a large complex of proteins I've characterized over the years when I was at University College London, which is found in the senescent cells. And upon their disruption, this complex of proteins restore fatty acid oxidation capacity in the T cell and therefore allows back the telomere transfer. But again, obviously, DOS is only one way to trigger telomere transfer, and telomere transfer is not DOS specific only, obviously. And so this is, what we have now under revision in a Nature journal, and essentially it goes, into the mechanism that regulates what goes beyond the immune synapse alone, because then what was really the discovery was that during the immune response, the T cells engaging in fatty acid oxidation was obviously getting these telomeres for themselves, but not the entire donation of the telomere transfer was not entirely confined to the lymphocyte. Instead, the lymphocyte would take, I would say, 50% roughly of this donation, and the remaining part of this telomeric material of APC origin would instead be released into the circulation. So that links, for the first time, telomere transfer and the longevity of the CD4 T cell to the longevity and the rejuvenation of the entire organism. And we call these a telomere river. And this is what a telomere river looks like. These are lymph nodes of, animals that have been immunized. And during the memory response, when we did histology of these are pieces of data that I think we did over 10 years ago. So this is a long story that we built upon. And you can see the memory T cells on the left that is sharing this, extracellular telomeric material in white. And then if you zoom out and you see these beautiful vessel-like networks and architectures, then it looks like there are punctated, stream of genetic material in the lymph node vessels. So we imagined, some people thought at the time it was an artifact. I didn't believe that. So we, considered the hypothesis that this was actually genuine piece of information, unprecedented, and that this material would flow into the circulation and would keep us young. And it turns that this material is evolutionarily conserved. Not only is present into the mouse plasma, is present into human plasma, and is present into plant xylem. So regarding human plasma, some reviewers said, how is it possible you didn't vaccinate people? Well, we don't have to forget that as humans, we are exposed to pathogens and immune responses do not need to be always evoked by external vaccinations. So we have memory population of T cells, and we believe that this memory population of T cells in the lymph nodes, when they are resting, they just pulse at low pace these rivers of telomeres to keep us young. In the plant xylem, this process beautifully is connected with photosynthesis. So you need light in order for these rivers to be detected even. So it looks like we are looking at a new biological particle that has been always with us and we never saw it before. And then we asked if fatty acid oxidation is needed to trigger telomere transfer and therefore to generate this river release from the T cells that had received telomeres from the APC. Can we regulate fatty acid oxidation to regulate and fine-tune telomere transfer and eventually, of course, river release? As a model, we used senescent T cells because that's what we are quite famous for studying, and because senescent T cells lose the capacity to undergo fatty acid oxidation. You can see on the left, the senescent T cells lose the key enzyme CPT1A, which is this fatty acid transporter important for its rate limiting step for fatty acid oxidation. When you interrogate immunological synapses on TIRF technology and microscopy, you can see how senescent T cells don't really form ordered synapses, but actually the CD3 in green is diffuse. They don't cluster into what is called the bullae of the synapse. This is really important because when you have this bullae of the synapse, Mike, Dustin, and I in the past showed that this is important because from this bullae, there is this trigger of TCR microvesicles, and these TCR microvesicles are what's needed to trigger antigen-specific telomere transfer. So in other words, we discovered also an entire metabolic pathway that regulates this clustering of the CD3 at the immune synapse. And so the pathway is quite complex, but we could summarize in saying when there is fatty acid oxidation, that limits ceramide accumulation into the senescent cell and activates an enzyme called ceramidase. This enzyme called ceramidase eventually produces a downstream lipid called phosphatidylethanolamine, or PE, which is the single metabolite responsible for the clustering of the TCR at the synapse. What does that mean? It means that, for instance, you can restore fatty acid oxidation in senescent cells by turning on CPT1A into the senescent cell, as you can see here, compared to a controlled senescent synapse. And this CPT1A expressing synapse is now undistinguishable from a non-senescent T cell synapse. Or beautifully, you can just feed for 20 minutes P directly to the senescent cell, and again, it will phenocopy the CPT1A effect. So this lies downstream the CPT1A cascade. And this is important because once you do this modulation, as I said, telomere transfer can be evoked on planar B layers. The reviewers didn't understand this during peer reviewing. It's quite sad because I published it exactly in Nature Cell Biology, the exact same methodology, and they wanted to peer review again that paper that was published in Nature 2 years ago, so that was disappointing. But anyway, once you interrogate this on planar B layers, it's quite cool technology because you can essentially do whatever you want to do to the T cell, modulate their metabolism, then interrogate the T cells on the synapse. Based on the metabolism, they will form good synapses or not. The good synapses have these bullae fully formed, and after 20 minutes, you can remove the T cells from the B layer, and now the TCR microvesicles will stay on the B layer. So now you can put the antigen-presenting cells with fluorescent telomeres that you labeled, and only when antigen is present in the picture, you will see these telomere release on the B layer. So then you can measure and visualize the vesicles being released once the metabolism has been fine-tuned optimally for fatty acid oxidation in the T cells. These are very sophisticated experiments that the reviewer did not understand. And that also, of course, corresponds to telomere elongation into the T cell. So obviously the T cell, when you do this into the immune synapse, this mirrors instead, of course, that I would say the synapse bilayer. When you do that in the immune conjugates, this mirrors exactly this telomere transfer donation. So these extend telomeres by roughly 3,000 base pairs, and this is roughly 30 times more what a telomerase can do. We believe that not all telomeres get elongated through telomere transfer. We believe that only ultra short telomeres are elongated by telomere transfer. And this is a key difference because we think that's where telomeres cannot go, because where telomeres are very, very short, the telomerase fails to anchor because they sometimes miss the sheltering capping, so telomerase cannot attach to the telomeres. And anyway, even if it was doing that, if telomeres would qualify as ultra short, let's say below, I'd say 2,500 base pairs, telomerase adding every turn of the round of division 100 base pairs and then losing it, it will still enforcing cells into a loop whereby ultra short telomere signaling cannot be fixed. So you need a reset signal. That reset signal only can come from telomere transfer, to our knowledge, because telomere transfer then essentially shifted the burden outside ultra short telomeres domain, because obviously that's back 3,000, 4,000, 5,000 base pairs. So that ultra short telomere is not an ultra short telomere anymore. A telomerase cannot do that. So we used proteomics because we wanted to understand whether these telomere rivers were actually different from the telomere vesicles, having established that fatty acid oxidation was needed for telomere vesicle transfer and eventually then river regeneration. In the circulation, we asked the question, okay, are they really different or this is just some artifact? So what we did very simply and directly, we took telomere vesicles or we took rivers into the circulation. So this is coming from human serum or the vesicle coming from human APCs, where we stimulate and we ionomize it, as we showed in the Nature Cell Biology paper. And we had, I would say, extraordinary finding that the proteomics is really different. The telomere vesicles are highly glycolytics, and instead the rivers are hypoglycolytic, but they are enriched for stem-like factors. Okay. So this is an important piece of information because obviously, once T cells receive telomere transfer, they undergo a stem-like switch. And so we believed, then we proved it in this paper, that the stemness component of the rivers is of T cell origin. And in fact, then in the paper, through various experiments, we proved that telomeric material is of APC origin. The glycolytic enzymes are removed by the T cells through a process of AV-like division, some sort of asymmetric division. And instead, the stem enzymes are contributed by the T cells. Lipids are contributed, I would say, 50-50 from T cells in APC. So there's, of course, some new biogenesis or recycling of lipid material. And you can do, of course, confocal imaging. You can see through confocal imaging again, even in humans, similar to mice, these rivers again have these structures that reminds what we saw in the vessel-like networks. And I was talking to people and they were like, Alessio, we never saw these structures in T cell cultures. And I'm like, how did you keep your cells in cultures? Oh, we took T cells, we give bees. I'm like, well, of course you wouldn't see that because in order to see rivers, you need to have telomere vesicles. And the T cell will need to elaborate those telomere vesicles to release them. So these structures, obviously, if people culture only T cells, they will never see that. So you need to give them telomere vesicles. You need to activate it to the T cells. You need to activate the T cells. And then you need, obviously, to collect the supernatants. This is important because in these reverse pictures I'm showing, there is no APC. So we take the vesicles from the APCs, we feed them to the T cells, we activate the T cells, and 48 hours we recover the supernatant, and then within the heavier fraction, because it's much bigger than the exosome-like telomere vesicles, you can observe the river-like structure. There's no secrecy about that. And again, very excitingly, they are depleted of GAPDH, and in the depletion of this image is wrong, so it should be GAPDH negative, not positive. We corrected in the paper, but it is still an older version. So in the GAPDH negative fraction, it's written GAPDH positive, but it's wrong. GAPDH negative fraction, those are enriched from stem-like enzymes, like for instance WNT or RANKS2. Vice versa, in the presence of GAPDH, you do not have the stemness enrichment. So again, rivers are hyperglycolytic and they are enriched for stem enzymes contributed by the T cell. They undergo, as I said, some sort of AD-like division, and you can see beautifully, you can use CPT1A in order to observe this cluster of CPT1A enzymes in one cell versus the other cell. So you end up one cell has more CPT1A, the other cell has lower CPT1A, and of course, the CPT1A bright cell we demonstrated is the T cell that eventually will release a river into the supernatant, and we believe it to be the T cell that also release the rivers, of course, in vivo. So you can see again through, we did experiments with telomere transfer probing by flow cytometry, same technology we already published and validated in Nature Biology a couple of years ago, and CTV tracking. You can see in the first cell division, then using also CD4 high and low markers, we isolated this population of stem-like T cells that are bright for CPT1A and that have reduced GAPDH expression, and prove eventually that this is the population that releases river in circulation. Excitingly, this river release depend on an exosome pathway, because if you use an exosome inhibitor, you suppress the relief in the circulation, the river release in the circulation. These structures can be recovered at 3000 G centrifugation, so it means that they are much, as I showed in the confocal imaging, they are much larger than exosomes that are recovered after ultracentrifugation and 100,000 G. They are released. Then we did experiments because the reviewer asked which populations are really really release them. So we did sorting experiments and demonstrated that the stem-like T cells, as expected, are the cells that upon telomere transfer also generate rivers and release river in the circulation. So our stem-like T cells are very important, CD4s are very important for our longevity. So if you can push those cells, you can push our fitness overall. So there's brand new function of the immune system. There was some work in Science, I think in 2020, showing that mice with defects in a mitochondrial protein, I don't remember which protein, mitochondrial protein, had premature aging. They observe reduced lifespan. They observed more inflammation. I think there were some follow-up experiments now, because when I saw that paper, I always thought that missed an adoptive transfer experiments. I think they did some more work, excitingly now showing that the phenotypes depend on CD4 T cells. But again, I think this is the first study really showing how the CD4 T cells and the immune system are responsible on the other side of the story, right? Because we don't destroy through genetic inhibition. I think the elegance of this approach is that we observe what happens naturally in our bodies and how the immune system is supporting for decades our youth, and probably explaining how we suddenly age at some point, because once this beautiful mechanism is unlocked, everything collapses. So the good news is that now we have, a new, I would say, overall view and broad map on how to intervene at various points. And we hope that other people can contribute and feed into this exciting new piece of biology we are sharing. As a reference, again, the telomere vesicle here, you can see they're contaminated by this GAPDH signature. And I insisted on GAPDH because, as you'll see later on, it's a key reference point that regulates the content of these particles. So as I mentioned before, we did this sorting experiment showing that the stem-like T cells are the major river producers. And then we asked this question in vivo. So is this really CD4 dependent in vivo? The answer is yes. Because if you deplete mice, recipient mice, do these experiments in which, going back, we have these APCs, we live label the telomeres of these APCs with these, live Tel-C probes, and then we either load these APCs with antigen, or we leave them unloaded, or we even block with anti-LFA1 the AD capacity of the T cell to undergo AD-like division, and then we inject this into recipient animals with or without the CD4, and then we interrogate a couple of weeks later if there is presence of readers, then we now can connect antigen presentation and CD4 to reader release in vivo. It's very elegant. And this is what we see. So when we actually load APCs and we would transfer APCs into an animal that actually has these CD4s in the presence of antigen and APCs, you will see this beautiful river signature. How we define these rivers? These are objects into the serum 3000 G that do not have GAPDH and that are, of course, present the telomeres of APC origin. All the telomeres we see here are, of course, of APC origin because this is the fluorescence coming from the donor APC we have transplanted, adoptively transferred into the recipient, right? So once you remove instead the CD4 from the recipient animals before the adoptive transfer, you will see on the bottom left, even in the presence of antigen, if the recipients are CD4 depleted, there's no reverse because the reverse are essentially whatever background was present in the naive. You have the same effect if the CD4s are present but there is no antigen. You have the same effect if you use anti-LFA1 to block, as I said, this AD-like division. So again, I think these beautifully connect antigen presentation, AD-like division of T cells, and CD4s. Of course, in these animals, CD8s are present, but CD8s do not generate rivers. I think it's really important to stress how CD4s have been overlooked in the past, I would say, decades, probably because we didn't have enough reagents to study them in terms of the telomeres and so on. But there's some kind of unique and amazing piece of biology that is only understood if we focus on CD4s. Even when asking reviewers, they were telling in various papers we have now under review, they were saying, oh, but you have CD8s, CD8s should be killing stuff, should keep us fit. And I always say a very simple observation. People with HIV, if they don't get their medications, they die. And they have very high CD8 signatures. So CD8s are great, but they're not sufficient sometimes, or actually always, to keep us fit, because if we don't have CD4s, we die. So CD4s are unique and fantastic. Again, what we did was to get these rivers from the animals, as I showed you here. And so we do the experiment 2 weeks later, we find these rivers, these are young animals, of course. Then we take these rivers and we transplant these rivers into old animals, 20 months old at the time of transplant, and then we leave them for 1 year. And we observe, excitingly, 1 year later, broad rejuvenation throughout the body. We measure various parameters, beta-galactosidase, P16, IL-6 producing cell, IL-6 in the serum, and so on, pathways of stemness. And what we observe is that only when essentially there was a river generating in the animals, this would be matching then into recipients. Another important point in these experiments, reviewers were asking, oh, but you're not transferring the same amount of vesicles. Of course I am not. And that's the point. I'm transferring the same volume because the point is that we want to show what is the concentration purposely of, the rivers and their effect. If I were obviously normalizing that by the number of particles, we will lose the point, right? Because our point is that only when CD4s are present and engage into an immune response, there is this river release. In circumstances where we deplete the recipients of CD4, so we block AD-like, or we don't put antigen, we will have less river, so we transfer less material, and that's the point of the experiment. Then we ask a different question, but kind of very important and related. And I think personally, this is the most important and exciting piece of the entire application of this story, because then we looked into this and said, okay, these rivers are GAP-pH depleted. I showed before that the GAP-pH depletion were also enriched by the CD4s of stem enzymes. Of course, in the paper, we have more controls to signify that. We said, can we bypass T cells entirely? Can we act mimicking the T cell effect, depleting GAP-pH through an siRNA, for instance, into the APC, then derive these vesicles with ionomycin? Can we generate what we call an artificial river? Does this have an effect? And it turns that these have actually an effect that is even more powerful than telomere vesicles. So here you can see these are the effects 1 year after transfer. So these animals are roughly 32 months old. And this is the effect of telomere vesicle, Tirna control, or artificial river, Tirna GAP-pH. We use, telomeres as kind of our reference point for, I would say, that sometimes we can call it the biological clock. So when you use telomere vesicles versus the reference T0, that would be, I would say, the average telomere length of the old animals at the beginning of the experiment. So when the animals are 20 month old, okay, versus the Sirna control, you will see that the Sirna control has some sort of buffering effect. So it somehow impedes further erosion of telomeres during the year of the experiment. The rivers, the artificial rivers, have a much more powerful rejuvenating effect, which translate also in a stunning difference in terms of the overall aspect, the vitality of the animals. I think we had some movies as well of these animals that are much more active, their posture is different, their coat is different, their weight is different. The animals are rejuvenated. And these animals eventually, you keep observing these animals eventually, in our hands at least, they keep living 4.5 to 5 years. They eventually die of natural death. We don't observe cancer. So you have this cool phenomenon. You see these young animals, they live longer. At some point, they just die. We expected them inside. We don't see cancers. We don't make a claim that this is completely disease-free, but we haven't seen disease. And at some point, they just die. So the CD4s, we again asked, okay, can we do the reverse experiments? Can we put CD4s back into an old animals? Do they evoke telomere transfer? So the reverse experiments, and the answer is that yes, they do, but they need to be young or they need to be rejuvenated. To rejuvenate, one of the tools our lab has is called DOS. The DOS is the disruptor of the ESMAC, so it gives similar or better results than young rivers. We haven't yet compared, I must say, we haven't yet compared the proteomics of the rivers of a young animal and the rivers of an animal that has received the DOS T cells. So there might be difference. We don't know that, and we are doing that. Instead, if you look into the composition of the old serum, you see two things. There's so many less rivers circulating or these particles circulating, and most of them are contaminated by GAP-TH. So suggesting that the capacity of T cells to purify these rivers from GAP-TH and therefore generate these vesicles from GAP-TH and therefore generate rivers is compromised with age. And then again, really controversial, but this is what we see. We take these rivers. These rivers can be transplanted into other old animals, and these rivers trigger lifespan extension. These can be done through the rivers. They can be done by adoptive transfer of the DOS T cells. It can be done through the GAP-TH depletion of the artificial rivers. I think some people have commented on the background controls. So the mortality, yes, there are some background controls dying earlier, but it's roughly 30% to my eye. I think people should focus on the fact on the divergence at the point of the interventions, which is 20 months. At 20 months, there is this diversion. Even if you exclude background mortality, you still have the lifespan extension. In the Sirna control versus Sirna GAP-TH, where you do not see this background mortality, you still have this life extension. So I think people should engage with the data, looking at the data for what they are. I anticipate we have other controls that have intact mortality up to 20 months, and there is still the same result. There is nothing exciting to be discovered about. And we just wanted to show that we had at the time, this preprint. In the final paper, there would be further controls, but the controls, the background mortality before the intervention have nothing to do with the intervention per se. The individual dots are not individual animals. This is what PRISM look like, and the animals are what we show in the figure legends of the preprint. We will add more controls, but I wanted to answer this because I have seen some comments about it, and I was quite surprised about it. So I guess, and I hope this will serve to satisfy them. So our model for telomere regeneration is quite simple when we strip off of all the noise. We have a T cell and we have an antigen presenting cell. When the T cell engages on fatty acid oxidation, it can cluster through ceramidase activation. It can cluster the CD3 on the bullae of the synapse, and this triggers telomere transfer from the APC. The telomere transfer from the APC proceeds, as we showed in NCD, the greatest sheltering, includes speeds up and so on, through a calcium flux in the APC. These vesicles contain ROT51, and when these lands into the T cell, there's two things that happens. It extends telomeres, ultra short telomeres in the T cell that undergo stem-like switch, and upon stem-like switch and AD-like division, one of the two daughter cells, CPT1A bright cell, release rivers that are depleted of GAP-TH because the cell themselves has somehow reduced the GAP-TH tone of the daughter cell, right? So you have the stem-like T cell that within the first division releases these rivers. After the subsequent divisions, these capacities are now lost. We have another paper in revision in Nature, so I can't say much about this, but there are some T cells, I would say, what I can say is that there are some T cells that are hyperproliferating and that have evolved this unique capacity to produce these readers and to keep us fit. And the overall outcome is systemic rejuvenation, increase of stem-like signaling, and in our hands, extraordinary life extension. Something also that I think we should train ourselves in thinking is that if you think about the gain that we get through telomere transfer, which is roughly 30 times what you get through a telomerase activation. It somehow surprisingly matches incredibly the gain that we have observed in our lifespan extension studies, roughly 20, 30 times more than what in general in the longevity space is observed by turning telomerase or by rapamycin or mTOR inhibition. Something also that I want to add, which is really important, we do not see mTOR activation in all T cells, quite the opposite. So I am a bit confused when people argue that mTOR inhibitors have rejuvenating effect, and I don't believe it to be accurate. I think that mTOR inhibition can slow down aging, but it does not rejuvenate the immune system. And this is really important. So I think we should fix narratives that command saying that rapamycins or rapa-orthologs can rejuvenate the immune system. They don't. Because if you look into the senescent immune system of humans, they do not have mTOR kinases. You cannot block mTOR because there is nothing to be blocked. And we should be open about it. We should have the courage to own this statement. Calorie restriction is something that excitingly has had also some similar life extensions effect, which makes perfect sense because of course calorie restrictions trigger fatty acid oxidation. So again, it supports this exact model. Obviously, this approach probably wouldn't command people to stop eating and still evoking similar effects, but we'll see, how it translates into humans. And this is the transfer biology. So I think we can stop here. So this is the transfer biology that relates to rivers. Thank you.
That was great, Alessio. So I have a couple of quick things. For the cause of death, I know you mentioned that there was no obvious pathology. I wonder, so in calorie-restricted animals, it's a similar story. Some of the calorie-restricted animals that have dramatically extended longevity, who have no obvious cause of death, it could be related to neurodegeneration, where the innervation of the diaphragm it denervates with age. And if you live long enough, it'll be like you have neurodegenerative disease. So I wonder, and I don't know how you would study this, but I wonder if maybe
but then they would act differently, right?
If they would die in their sleep, you wouldn't even know.
Yeah. No, I mean, they would act differently during their life, right? In the end stage of the life, probably. They would move differently, maybe.
Yeah, that definitely could be possible, especially if there's lower and lower levels of the diaphragm being innervated. But it's possible that you should see a distribution where, maybe the ones that are the most highly innervated, you don't see some obvious phenotype, but the ones that are...
I didn't look at this at all. It's very interesting, but I didn't look at this at all. So
but it raises the question.
We stopped at that. Yes.
No worries. You've done so much, and I'm grateful for your contribution to the field, eternally grateful, because like I said, this is huge. But I wonder if maybe the telomere rivers aren't rejuvenating something, right? Or you've reached the natural capped maximum lifespan in a mouse up to 60 months, and there's just nothing else you can do. Whatever.
I think that's a plateau because we don't see a lot of difference from the natural rivers and the artificial rivers, although the phenotype differ. And I think you had a questions on that as well. The answer to that question now that I have in mind is that very simply, if you think about, the fact that T cells uses roughly 50% of the telomeric material for themselves, and instead with the artificial rivers you bypass the CD4s entirely. So when you measure that, you know that you have roughly twice the amount of telomeric material and of course stem enriched enzymes into the artificial river, so they are more powerful because of that.
Yeah, definitely. The people who are saying about short-lived controls, sure, your controls were on average, it was probably around 25 months median lifespan. But in those studies that have short-lived controls and they give some intervention, they'll get the intervention to only 30 months, which is the median lifespan of C57.
Exactly.
They didn't do anything.
Exactly.
But
it's not that they're going to live at 5 years, right? Even if everyone was living at 20 before the intervention. Exactly.
Yeah, exactly. And yours didn't get to 30 months. They got to 45 months median lifespan. And even the 10% survival, the 10% survival of the controls wasn't terrible. Or it was close to 40 months, which is what you would expect in C57. So maximum lifespan, 10% survival in the controls was pretty good. And you got your telomere rivers treated, blew way past that. The antigen presenting cells, they're responding to the antigenic stimulus, which in your study you used, like influenza, I think was one of the antigens that you used. So is the antigenic signal so it could be flu, it could be COVID, it could be any virus, any bacteria that the APCs present to the T cells, and then you get the telomere rivers. But how important is the antigenic signal? Is virus different from bacteria? Or any antigenic signal that activates the APCs will then lead to this mechanism of the T cells and telomere rivers being, released everywhere?
So that's a good question. So the mechanism is, of course, requires antigen presence, and we didn't really stratify by antigens, but I think by gut feeling, the answer would be that obviously this depends on a stable synapse. So if you have strong antigens that induce a stable synapse, you will have more TCR microvesicle release from the synapse and thereby more river release.
So it could be your guess or speculation would be it could be any antigen, virus.
Yes, it is surely any antigen, but in terms of the magnitude of the process, strong antigens will elicit in the presence of fatty acid oxidation, stronger release.
Got it. And then, so I wonder if this is an evolutionary process aimed at restoring systemic homeostasis after an infection, right? Because
Absolutely.
when you think about infection, you're going to have oxidative stress, immune activation. Oxidative stress is a part of controlling the infection. So the APCs, like I said, are are part of this evolutionary process where it's like, all right, we've been wrecked temporarily. We need to restore. But then I'm guessing, I don't know if you've looked at it during aging, where the APCs are less able either to form the T cell connection during
Yes.
aging, and
Yes.
then maybe
Yes.
even because you've got FAO, fatty acid oxidation defects during aging, that probably the efficiency of the transfer is also going to be down during aging, and then it's just
Absolutely.
yeah. So
Absolutely. Yeah, we show that. And even if you think about the senescent cells that accumulate during aging, the senescent cells don't do this very well. So, and I believe that the shift that you have during aging into the myeloid compartment, it's a mechanism that tries to compensate for the lack of efficiency of this process. So physiology, as you know better than me, is all connected, right? So if you apply this lens, then you have this beautiful picture. So the T cells are less capable of evoking telomere transfer, and therefore the body try to compensate, shifting towards the myeloid lineage, but still is not doing a very good job in doing that.
yeah. So then it raises the question too, if you did one transfer at 20 months and that pushed it out like another 20 months. So maybe there's an optimal frequency for is once sufficient? Maybe you do this, the classic Aubrey to
But
Gray.
even in animals, when we have, because the same phenomena is also evoked by, of course, a DOS injection per se. So if you are animals on DOS, live at least for like 4 years. And we also did repeated injections of that. And somehow always they die between 4 and 5 years. So
Wow.
we have a solid, I would say we have a solid 1 year plus at least. And I think they reach some sort of plateau around 4.5 to 5 years.
Yeah. So it doesn't have to be the rivers as long as you're inhibiting the SMAC complex. So
Oh,
then,
yeah, yeah, of course. Yeah.
but then how often would that frequency need to happen? Would that need to be a daily
With the
thing?
DOS, only one injection.
Wow.
But we tried also multiple injections, and we have similar results. So that's why I'm saying, like, more blocking is the rejuvenation biology is really what we observe. That the rejuvenation biology is a cascade of events. Even people were saying, "Oh, how is it possible that you transfer 5,000 vesicles and you have the systemic effects?" Then I will say, "Well, guys, have you heard of hormones that works in picograms and control the entire body? This is very similar. We have ideas of why and how signal amplification occurs, I cannot release now, obviously, but signal amplifications with how biology works and physiology as well. So obviously, the 5,000 vesicles are, the vesicles that in our hands are sufficient to trigger the rejuvenation. For the rivers, even less than that. Sometimes 2,000 vesicles for the artificial rivers are more than enough.
And then when the APCs rejuvenate the CD4s, is
Yes.
telomere length in the old animals the similar length than it is in the young, or it goes beyond that?
It is towards the young, not at the level of the young, but towards the young reference, I would say. I
So
would say
5,
60, 70% of the young reference.
and then without it, it's probably what percentage? Like the telomere.
Well, the without it will be like without the transfer. The difference between old and young probably is like half of that length. And once you restore it, they would go to, the difference would be like 10%, 20%.
So not all the way. Is that also true with the other tissues that are rejuvenated by the telomere rivers, or
Yeah,
is
I'm talking about the various tissues here.
so it seems like there's actually some room to push it out even further to get maximum restorative effect?
But here again, I am talking about a single transplant. I've not done repeated transplants of rivers as yet.
Are there side effects of the river? I mean, granted, the rejuvenation effect is an improvement in or reduction in senescent
I
cells.
do not see side effects. And even for the DOS, which we are launching a clinical trial now, the characteristics of it is that up to doses which are hundreds of times the therapeutic dose, there is no side effect. So you have the signature where the NOEL overlaps with the MFT. So the only limit is the solubility of the compound. And you can push it 600 or 700 times the therapeutic dose. And of course, there is transient inflammation at the site of injection and stuff like that, but there is no long-term effect.
That's amazing. The SMAC, right? So the SMAC becomes more active during aging, but what's
Yes.
triggering the SMAC? So, is the SMAC accumulating during aging because of chronic antigenic stimulus throughout the lifespan? Or is it known why SMAC?
Okay, we have a paper in Nature in revision describing exactly this. So I cannot tell you exactly how an SMAC is formed. So I will keep it high level. The SMAC is formed when there's proteins called the sestrins, and this protein is part of a stress response. And in certain circumstances, I would say, this protein, instead of binding mTOR, which is what David Sabatini had shown, where they have an anti-aging function, they bind to MAP kinases and they have a pro-aging function. Your question relates how the SMAC is formed. Unfortunately, I cannot answer because the paper is under review in Nature and I haven't released the preprint on the bioarchive.
Yeah, not necessarily how it's formed, but what's leading to an increase of its formation during aging. Is that because of chronic microbial burden? The mice just accumulate.
We have shown that there is an immediate cascade that leads to its formation. But again, I cannot tell you how, because otherwise I will leak out the know-how.
No worries. All right. And then, so calorie restriction, right? So I wonder if, do you have plans on looking at whether CD4 T cells, so the efficiency of this process would be better in aged CR animals relative to non-CR ad lib animals?
I'm sorry?
So if you take a calorie-restricted mouse and an old CR mouse and look at the efficiency of the APC CD4 rejuvenation and then systemic telomere release and rejuvenation, maybe you get an even greater effect in quote unquote healthier animals for a given age. Do you have plans to look at that?
So you mean, if I understand the question, you mean that the liver transplant might be more efficient under the calorie restriction regimen? That's what you're asking?
For a given age, yeah.
Could be because
Yeah.
it amplifies background fatty acid oxidation. It could be interesting to look at it. I haven't done it, but yes.
Right, but calorie-restricted mice, they're not immortal too, and fatty acid oxidation in T cells and other organs, mitochondrial function will decline with age too. So,
Yes.
but I wonder if, if you've got a calorie restricted mouse that lives average and maximal 40 and 50, and now you give them a rejuvenation, telomere maybe not at 20 months, maybe you give it to them later at 30 months even. Does that push
Hmm.
them out to 7 years or 6 years?
Yeah,
That
I
would
don't know
be,
yet. That would be
yeah,
amazing. But
yeah.
at least we are learning that the limits, I think what is important, the message that we should give, in my opinion, is that the limits that we impose ourselves to and the constraints we impose ourselves to are dictated by the fact that we only add half of the picture of the story and we confused anti-aging with rejuvenation. So that's a big problem in the field. And a lot of skepticism arises exactly from here. Even, supporters of mTOR inhibition will say that rapamycin has some effect. There's a Nature paper. I know the paper. I know all the papers, but the point is that we need to look at it honestly. And rapamycin, of course, they've shown in Nature that has effects also later on in life, but the effects are lower in a younger age and they are quite mild, I would say. So mTOR inhibition is something that delays aging, and progressively the capacity of mTOR inhibition of delaying aging declines with age because mTOR signaling is lost with age.
Rapamycin, fungi are a normal part of the gut microbiome. So I wonder, in fungi, translocation from the gut into the blood increases during aging. In support of that, the incidence of hospitalization with fungemia in older adults, it's like 10 to 12 times higher in old versus young. So I wonder if most of the rapamycin story, even though the mice are SPF, but still you've got the mice have fungi in their gut. So I think it's plausible that the rapamycin extension of lifespan is just basically ameliorating. And there's no data. Nobody's looked at this, which is even more sad. So I'm just speculating. But I wonder if rapamycin is doing some kind of an antifungal effect, not a
It's
rejuvenation.
possible. It's
And that
possible.
and that's the
And in the
extension.
end, it's some sort of antibiotics, right? So why not?
Yeah, but I'm with you. So much of the field is so hot on rapamycin and all these other anti-aging stuff, but with the exception of
It's
calorie.
terrible.
Yeah.
Calorie restriction is different though. It's a different thing because actually, it triggers independently of the mTOR presence, it triggers metabolic remodeling. So what we are questioning is the fact, why would you use an mTOR blocker for a kinase that either you tell me that rapamycin is not acting on mTOR, and then you gotta characterize the mechanism, or if you have to look at the data that we have and we've been having for like the last 15 years, it's no mTOR to be blocked. So I don't know what they're talking about.
Yeah, nice. Are there other things in the rivers containing vesicles? Like, have you done a metabolomic profile? I think you mentioned you looked at the proteomic stuff, but
Proteomics, yes.
yep.
We've done proteomics. I think we released already also the raw data on it. And most of it are, well, of course, we chose what we wanted to show for the story. But, on the top of my mind, there's a lot of stuff for vesicle communication, of course, cytoskeleton stuff as well. And but the most striking thing was the downregulation of glycolysis and upregulation in stemness.
Yeah, well, that's essential for the rejuvenation effect, right? So
Yes, yes,
yeah, but I
yes.
wonder if there's other stuff in the vesicles besides the rivers, besides cytoskeletal machinery, maybe even like translation, transcriptional machinery related things, but maybe
I
metabolite.
don't remember. I don't remember. It's the data is online, so but I don't remember. So I cannot tell you because I don't remember if there is something else right now.
Nice.
But
All
there
right.
should be some other stuff, of course.
Yeah. Current, future studies, things you can say that you're working on versus.
Well, we have right now we have three papers um, under review in Nature-like journals, I would say, and actually two papers, another one in submission, but hopefully in Nature as well. Um, this is the river. This is one paper. Then we have a major paper on the platform of the DOS where we present this paper. This paper has done a long journey. So I think you mentioned the 8 months. The general peer reviewing for my papers is 4 years. So this paper, or the telomere transfer paper, was reviewed for 4 years before getting out. The DOS also 4 years. And hopefully, fingers crossed, is going to be now done. Again, I think he has done 18 different reviewers, 18, and across 5 or 6 different Nature journals. So it's it's a lot of reviewing, I would say. And then we have this other very exciting shorter paper. Don't know if you've seen it, on the HIV functional cure. We released, I think, a month ago, and where we showed that as a byproduct of the rejuvenation response, you trigger these drug enzymes, which in the context of HIV infection compete with HIV integrases to chop off the proviral DNA. And in many people, we observe HIV negative state within hours of treatment. It's amazing. It's
Nice.
great. As you said at the beginning, the story is so cohesive. If you look at what we have put through the last 15 years, right? So either this is a big hallucination that I've got for the past 20 years, or there must be some truth behind, because it's quite significant, the fact that it's all connected. We start with the SMAC, then telomere transfer. Now we show that if you block the SMAC, you restore telomere transfer, and the telomere transfer then trigger reverse, and then this transfer biology cures HIV. So it looks a very cohesive story from the onset to the link with physiology and rejuvenation, and now clinical applications. So I guess that once we then now get into humans this year, and we show hopefully transfer signature in a human, once we give DOS and a vaccine, the skeptics will say, okay, this was just new, and that's it, and move on. And like, science is done by discoveries, right? That's what we should be doing. We should be discovering things, and we should support people who discover things, because it's a beautiful but also very painful journey. So, and we do this to share it with everyone. So that's what we want to do, right? We want to share it.
And they're slowing it down. Yeah. So in terms of how you envision, this translating into people, I mean, granted, it's got to go through clinical trials, but in translating, like, how would you foresee? So someone shows up at SenseCell, and then so that's your company for people who don't know, but they just show up and you give them, I'm knowing that it can be an artificial reverse generation, or do you have to do an adoptive transfer? You pull their T cells out, and then, so how do you foresee this playing out in terms of being able to promote human rejuvenation?
The first thing that we will be doing very soon, because the DOS clinical ready, GMP ready, is to be released in a month or so. So we're starting trials at University College London, most likely this year. We recruit people on the base of their pharmacodynamics, therefore from the blood. It's not anymore, unfortunately, senescence is not a matter anymore of age. So you have after COVID, you have a lot of immune senescence for a variety of reasons I don't want to discuss, but you have a lot of immune senescence, and this spans across lifespan. So we see accelerated immune senescence in people who are 20 or 25. And profiles, when I started working on immune senescence, were characteristics of people in their 70s. So this tremendous and dramatic acceleration to immune senescence is something that is spanning through our life. So obviously, in the context of a clinical trials and a rejuvenation effort, we don't have anymore just to select based on age. What we're going to do, we're going to recruit people, of course, based on the medical history, but even regardless of age, taking the blood and observe, do they have senescent cells? Do they have SMACs? And we have parameters to determine whether, for instance, if you have, I would say, 30% of senescent CD4s in your peripheral compartment, that's pretty bad for you. And we see these very often nowadays. in people who are 30 or 25. And in 2014, when I was doing my PhD, these were phenotypes of people in their 70s or 60s. So there's an acceleration in immune senescence, and really our data are, we could say that there is a before and after 2019. So before 2019, there was a certain trend, and afterwards a completely immense shift and accelerated immune senescence. So these must be addressed in the clinical trials, and they will be part of the recruiting. DOS injection is the first treatment that we have available, because the treatment in which we invested most. And this is a systemic injection, so there will be an intramuscular injection, beginning with one injection. In the context of severe immune rejuvenation, then of course we plan on doing more than one injection. But even at one injection, we expect to see the transfer biology and a significant rejuvenation of the immune system. If that also, as a bonus, triggers systemic rejuvenation, that's not under the remit of the trial, obviously, because as you know, MHRA and FDA do not consider aging a disease per se, but obviously, we will measure liver biology, and we will record whether people felt less sick or not, as we expect, less infections during the years or not, and the transfer biology that we have described as a primary outcome that we can verify within, I would say, days or weeks from the onset of the trial. Once we give a vaccine and the DOS in the people compared to the control, we expect to see the shift in stem signatures in the immune system, which is very exciting, the rejuvenation of the senescent immune system, which is very exciting, the disruption of the asthma, which is very exciting, and the river signatures, which are also very exciting, circulating in the serum. So that answered part of your questions. In the future, I think there's a lot of space for having medicines in the forms of, for instance, artificial readers, whereby people could get some of their blood could be modified, for instance, modulating and removing FDH from the vesicle preparation, and this GMP vesicle preparation re-injected in the people. So I don't really think that we will do adoptive transfer of cells. I think those will be overtaken by curing people with their own blood prep that have undergone modifications or their vesicle-like components. Vesicles are amazing. The only issues with that is that you need to be, there are constraints into the GMP purifications of them that under GLP must be tightly regulated. But if you have the capacity of doing that, they are very powerful tools. So,
Nice.
yes.
So are there things you're doing aside from DOS injections, that like lifestyle related things that people can do until we get to the point, like that can help?
Yeah, well, I guess, of course, physical activity, right? And moderate physical activities control insulin spikes because they activate mTOR, and mTOR activation is somehow linked to asthma generation. Yeah, that's it. And I guess that it wouldn't be safe for me to tell people what sort of foods or supplements to take to suppress these pathways.
And then two more quick questions. So
Sure.
so because we can't measure senescent T cells, it's not commercially available. It's just we can measure CD4 levels or CD8 levels, but that doesn't tell you about functional status. So when considering that the SAS part of that is IL-6, TNF alpha, granted there are more pro-inflammatory cytokines and other proteins that are
Sure.
produced, but if you look at IL-6, TNF alpha just as an indirect proxy at various time points throughout the aging process, granted those pro-inflammatory cytokines increase during aging. So could that be used as an indirect measure in the short term for measures? It wouldn't.
We see, especially IL-6, at least in mice, we tried them all, and we see that especially IL-6 among all the cytokines, IL-6 seems to be the one more reliable for mirroring, I would say, the effect of the reverse. TNF-alpha and interferon gamma, the effects are a bit unstable, so not really sure. But the issue, I think the issues with this is that, you know, you need to be extremely careful in understanding, you know, who is the contributor of the cytokines. Because I think this is a problem of the field of immunology. When they just use the cytokine, they don't know who is producing that. And because if you observe, you know, they sometimes, you know, often they do interferon gamma positive CD4s or TNF-alpha positive. Okay, it doesn't mean anything to me. Like, are they effector T cells? Are they senescent cells? So are the cells that are releasing that as a genuine part of an antigen-specific response, or is that part of inflammation? So I think you must incorporate phenotype into the picture. And as you said, it's currently missing. So I think it's really important and exciting to incorporate those sort of markers, C27, C28, negative CD4s or CD8s, and measure what's their contribution, you know, in terms of intracellular subs signatures, for instance, right, in the peripheral blood. So refining these, because I think what is really important is that we have to have something which is very neat, so that then can be used in order to monitor progress, and rather than just relying on you know, proxy markers of ambiguous or promiscuous origin.
Definitely. So CV27, CV28 is a part of that. That's not commercially available either. So now we're back to that. How can we,
Yes.
which is, you know, that's a place where maybe there's something that can be offered as that commercial, you know, a commercial service.
Absolutely, absolutely.
And
And
then
we will do this in our trials, for instance, for sure.
fantastic.
For us is usual, is normal, because we've been doing this for 20 years, but no one does it, right? So we will be the first company that actually look at the blood, look at these cell populations, and can tell you what's the age of your immune system, regardless of your chronological age, sorry, and then include you or not in a DOS or rejuvenation trial, right? A big part of the future is that regardless of vaccination, if you take on a DOS injection, your immune system is reprogrammed and reset. And therefore, it is in a readiness state. This is what the DOS paper in Science Division is about. It's in a readiness state whereby you or the animal doesn't have to make a prediction based on vaccination of what the animal or the individual will catch in the future. Right now, our current model in immunology is based on training the immune system through vaccines, right? So based on whatever challenge you expect to get or to be faced with, you design a vaccination campaign, for instance, influenza every year, COVID, and so on, right? In terms of, and of course, you know, the repeated vaccinations, they narrow the repertoire because they expand the antigen-specific populations, and over time, of course, the TCR repertoire shrinks because of the exposure over lifetime, right? With the immune reset, what we do is completely opposite. You can couple it with vaccination, of course, but and I think the most exciting part of this is that you can completely remove the vaccine from the picture and forget for now about the reverse and say, okay, I'll give you just an injection. You won't see immediate telomerase transfer because there's no antigen there. But what you do, you remove Fmax from the picture, and you'll trigger the stem switch. And therefore, those cells, once an antigen will come, they will make sure they sustain an immune response towards the antigen. We show that this is based on a process of TCR revision. I'm not sure whether you've heard 25 years ago, people like Antonio Lanzavecchia, they were describing this process of TCR revision in the periphery. They didn't really understand how. Sometimes the RAC proteins were expressed, and this led to, even in mice, they did some model with, I think, Pamela Fink did it, showing that there were some models of peripheral TCR revision, didn't understand how. And in the US paper, we showed that's a consequence of asthma disruption. So when you remove these metabolic stress pathways from the T cells again gain the sort of flexibility to, re-express and modify the TCR beta gene. There's no autoimmunity in the process because the T cells with the new TCRs, we track them back. They go to the thymus to again thymus checks and selections. So zero case of autoimmunity at all. But the beauty of these is that we sequence the repertoire, DNA sequencing of the repertoire, and they have new beta pockets, and these beta pockets are cross-reactive, so they can recognize, conserve epitopes that are present in yellow fever and in ABB and in CMV. So they offer a broad spectrum protection. And I think that the future of immunology is based not on trained immunity, but on licensing the immune system itself, the ability of recognizing new challenges that you can see in the future. Think about cancer vaccines. Sometimes cancer vaccines try to do that, right? Predict what sort will be the signatures of the cancers. Here we're saying we'll go beyond that. We'll reset the immune system. We'll make sure the immune system is into a metabolic state based on fatty acid oxidation, of course, that whereby the antigen is present in the future. So months later from the US injection, you will be ready to face it without any vaccine. And this is what we've done in the US paper. Like we gave it to animals in the one which is in final review, gave it to animals, and 6 months later, the animals can get infected with flu without any vaccine, and they live without the vaccines. And if you vaccinate the animals without the US, the old animals, they die because the vaccine is not very effective at old age. And this is dependent on CD4. So you can take the CD4s and transplant them back, and you see that the coverage. So it's entirely dependent on the TCR revision process outside the thymus. That's very exciting, and I think it really shows some new way of approaching immunological challenges in the future. Think about pandemics and stuff like that, right? You could program your immune system to be able to face things we don't even know what they will be, but you know that your immune system is into good health and therefore capable of facing those challenges.
And I don't want to say it's as simple as, but it's as simple as taking the SMAC inhibitor in order to give your CD4s even that possibility of having that broad.
Yes.
Yeah,
Yes.
amazing. A perfect place to end here for now. I have so many more other questions,
Yes.
but I'm looking forward to obviously I'm looking forward to more coming from your group, and hopefully we can get
Absolutely.
in the next few months and dig deeper.
Absolutely.
So yeah, great, Alessio. Talk to you soon.
Thank you. Thank
Uh,
you.
all right.
Bye-bye. Thank you.